Okay, good morning all students. I'm going to start my lecture and it will be a very short lecture. As I told you yesterday, I'm just going to cover the chemoautotrophs, in which I'm going to cover today iron bacteria. Okay, good morning again to all students. Various freshwater ponds, springs have high content of iron salts. Okay, and it has been known that the distinct bacterial flora are associated with such habitats, and these iron bacteria form colonies that are heavily encrusted with ferric hydroxide. However, since most of the iron springs are neutral or alkaline, ferrous iron undergoes rapid spontaneous oxidation, so it has proven very difficult to ascertain the role, if any, that of the iron oxidation plays in the metabolism of the bacteria. The most conspicuous iron bacteria, most conspicuous iron bacteria are filamentous cythid, okay, of one of the well-known group in this is the Sparotilus group, in many of which sheaths are encrusted with the iron oxide. Sheaths are encrusted with the iron oxides. Okay, and they can be readily grown as heterotrophs also, and can be isolated in pure culture. But there are various others which can be grown chemoautotrophically. Okay, which we will study now. Although it is possible to show that such pure cultures accumulate iron oxides on their sheets, and there is no evidence that such deposition is physiologically significant process, or that this bacteria can develop as a chemoautotrophs. Okay, they are just considerations, it may be possible. Okay, so one of such bacteria, okay, is Gallionella. All attempts to obtain the cultures of the Gallionella in organic media have been failed. Okay, but it has been grown in the mineral medium containing ferrous sulfide, and the use of virtually insoluble ferrous salts minimize the rate of spontaneous oxidation under neutral conditions. In these cultures, Gallionella will form cottony colonies attached to the wall of the vessel. Okay, so the term obligate autotrophy, okay, obligate autotrophy, some undergo obligate autotrophy also, but there are various assumptions regarding autotrophy in the iron bacteria also. Okay, so now again discussing about the Gallionella. Much of the colony is an inorganic, small bean-shaped bacterial cells which are located at the branch tips and are gram-negative, microaerophilic, chemolithotrophic, okay, and can secrete a twisted stalk-like structures. You may have studied stalk bacteria, okay. Yes, you have not studied stalk bacteria, sorry. It was not in your syllabus, but the new syllabus of the Saurashtra University have included stalked bacteria, some of the bacteria, okay. If you want to study, you can join their lectures also. I will tell you when there will be a lecture on stalked bacteria, okay. And can grow with a ferrous iron as a source of energy and the carbon dioxide as a sole source of carbon. Okay, so the obligate phototrophy, as you know, is basically an expression of the extreme specialization and adaptation to the respiration of inorganic substrates, and several hypotheses are given with regarding to the autotrophy in the case of Gallionella. Okay, and these Gallionella are generally found in drain pipes, mountain streams associated with the flakes and thick coverings of iron oxides. Until a few years ago, it was not known whether they use energy or not in the oxidation of Fe2 to Fe3+. Okay, and thus we can assume they grow autotrophically. And quite recently, it has been demonstrated uh, that the Gallionella are able to use carbon dioxide through the ribulose biphosphate carboxylase. Okay, so we can now conclude that the Gallionella are basically lithoautotrophic bacteria. Okay, more specifically lithoautotrophic bacteria. Okay, and similar another group, okay, another bacteria which is also a sheathed bacteria, okay, is an Leptothrix discophorus which can oxidize manganese, okay, which can oxidize manganese. Okay, so it depends on the type and the system, even the species of the bacteria. And some of the nitrifying as well as sulfur bacteria have a branch respiratory chain. Okay, and this may lead to interlink between various and various iron bacteria. Okay, and one of the most studied iron bacterium, okay, is basically Thiobacillus pyroxidans, which oxidizes ferrous to ferric iron. Okay, ferrous to ferric iron. Okay, and it can tolerate pH values of about 2.5 also. Okay, it can tolerate pH value of about 2.5 also. Okay, and their cell diameter is about 1.2 to 2.5. Okay, 1.5, sorry, 1.2 to 2.5 micrometer, and a GC percent ratio of 69 to 71%. Okay, which is basically an identification feature. Now moving forward, okay, now moving further, how basically they work. Okay, now Fe2 is used as an electron donor in some gram-negative bacteria including Gallionella, Ferruginea and Thiobacillus pyroxidans. And this electron transfer involves basically a rusty cyanine. Okay, what is rusty cyanine? A copper containing blue protein, and is not involved in the electron transport chain that oxidizes sulfur compounds in this bacterium. Okay, so if you want to write the definition of rusty cyanine, okay, you can write just it's a copper containing blue protein which is involved in the oxidation of sulfur compounds or iron compounds in a short. Okay, and this question is asked many times, at least 3 or 4 times in last 5 years, at least 3 or 4 times in 5 years. Okay, in one, this is 1 mark question. Okay, and also the free energy change and redox potential is similar to that of oxygen, and the acidophilic oxidizers maintain their internal pH, and they maintain a low or inside positive membrane potential to compensate for the large potential generated by the hydrogen gradient. And the electron transfer from the periplasmic region to the cytochrome oxidase contributes to the membrane potential and the proton consumption by the oxidase, and which contributes to the proton gradient part of the proton motive force. This is an overall diagram, okay, how ATP synthesis coupled to the oxidation of Fe2 plus in Thiobacillus pyroxidans, in which two main enzymes, okay, sorry, two more electron carriers or acceptors. One is rustacyanine, it's in blue colored protein, and second one is a cytochrome oxidase. And the ability of various acidophilic iron and sulfur oxidizing bacteria is to convert sulfur oxides to water soluble heavy metal sulfates, okay, and is utilized for the leaching of ores, okay, of the low grade ores, more particularly to obtain copper, nickel, zinc, molybdenum, and even uranium, and even uranium. Okay, so this is basically a leaching process. Okay, this bacteria can help us in the leaching process of the various ores. Okay, and various metals are obtained by concentration and the precipitation. Okay, so in such processes, heavy metals into the solution is the result of several associated processes. As I told you earlier, that sulfur and iron bacteria are related to each other with regard to the processes. Okay, so bacterial oxidations of the reduced sulfur compounds or elemental sulfur, which convert sulfur, elemental sulfur, to the sulfuric acid and ferrous to the ferric. Okay, and also by the chemical oxidation of insoluble metal salts to the soluble metal sulfates and sulfurs. Okay, so one of such reaction is the conversion of FeS2 into FeSO4. Okay, and another one is conversion of elemental sulfur into sulfuric acid. Okay, so basically the bacteria are responsible for the supply of sulfuric acid and for the respiration of ferric, and both the components are consumed in the solubilization of the ores. Okay, and these conversions are carried out by the thiobacillus thiooxidans as well as thiobacillus pharioxidans. Okay, so the strains involved, okay, are basically tolerant to various heavy metals also. Okay, so some of the other strains which can oxidize iron is an most popular one is an sulfolobus. It's an sulfolobus. Okay, now again back to the mechanism. Okay, first mechanism with regard to sulfur, which in which elemental sulfur will be converted into H2SO4. Okay, and FeS2 will be converted into FeSO4. Okay, now these both combine. Okay, these both combine FeSO4 and H2SO4 combines and form Fe2SO4 thrice. Okay, in which they can be converted. Okay, so in the cytoplasm you can see the pH is around 6, outer membrane. Okay, pH may be around 2. So this bacteria maintain pH in their internal part, and the rusty cyanide here in this case, okay, where cytochrome C2 will be used, okay, which will be reduced and oxidized. This will be reduced and oxidized. It helps in the conversion of Fe2 into Fe3. Okay, in this case rusty cyanide will be used. At the same time, hydrogen will enter the cell, okay, in the cytoplasm and help to generate energy generate energy. Okay, and in this case cytochromes basically pass on oxygen. Okay, and with the help of cytochrome oxidase and further other enzymes, oxygen will be converted into water molecule. Oxygen will be converted into a water molecule. Okay, I am repeating the whole process again. Okay, the ATP synthesis is coupled to the oxidation of Fe2 into Fe3. Okay, in which two main compounds are involved are rusty cyanine and cytochrome oxidase. Okay, the place at a time. Okay, Fe2 plus will be converted into Fe3, that is ferrous into ferric. Okay, with the help of cytochrome C2. Okay, with the help of cytochrome C2, okay, a cytochrome will be reduced. Okay, and this reduced form will be converted into cytochrome oxidized form. Okay, and with the help of rusty cyanine, oxygen is then transferred to the cytochrome oxidase where it forms water molecule. At the same time, the hydrogen gradient which is formed in this case will help to produce the ATP molecules. Okay, and you can see clear difference in the pH, where pH in the periplasm is 2 and that of cytoplasm is around 6. Okay, so these are the photographs and one of the important reactions and final reactions, okay, where Fe3 can be further used by the bacteria. Okay, so this is an example of an acidothiobacillus peroxidans. Okay, and this is an example photograph seen here is of an acid mine drainage. Okay, this type of drainage is seen usually around the mines. Okay, where acid drainage is one of the most one of the most important problems, rather than not important, but various notorious problems, okay, in the mines. The acid mine drainage showing the confluence of a normal river and a creek draining of a coal mine area, and at low pH values Fe2 plus does not oxidize simultaneously in air, but the bacteria Acidothiobacillus peroxidans carries out the oxidation, and the insoluble FeOH3 and the complex ferric salts are basically precipitated. Okay, are precipitated, giving to a red coloration, giving rise to a similar to that of a something like red coloration. Okay, when these cultures of Acidothiobacillus peroxidans as is shown in this series, okay, shown in dilution series with no growth in the tube on the left, you can see clear there is no growth. Okay, as we move forward, okay, the growth basically increases, okay, as it's a dilution series. Okay, so growth is evident by the production of FeOH thrice. Okay, now you can clearly see in a darker shade, okay, which is produced by FeOH thrice. Okay, and with the help of ferrous oxidants, okay. Now moving forward, okay, now this is just an ion cycle, okay, and easy one, simply if you want to remember. Okay, and aerobic, under aerobic conditions, under anaerobic conditions, which are the bacteria which are involved, okay, which are involved in the ion respiration. Okay, in the case of uh, aerobic one, where Fe2 will be converted into Fe3, okay, and Fe3O4 will be converted further into Fe3. At the same time, Fe3 can be also converted into Fe3O4 under aerobic conditions. At neutral pH, Gallionella species is involved. At acidic pH, Leptospirillum and Thiobacillus peroxidans are involved. In acidic and thermophilic conditions, Sulfolobus is involved, which is an thermophile, Okay, now one of the bacteria under anaerobic conditions, which is also known as a type of a magnetic bacteria, okay, where it converts Fe3 into Fe3O4, okay, Fe3 into FeO4, okay, and bacteria is Aquaspirillum magnetotacticum. Okay, I'm repeating the word again, Aquaspirillum magnetotacticum. Okay, and in this case you can see a purple photobacteria also involved. Remember the Vinogredsky column. Okay, remember your Vinogredsky column, where purple photobacteria, in the layer of purple photobacteria you can clearly see the red patches of iron oxides, okay, and all this happens in the case of anaerobic conditions. And other bacteria which are also involved are Farebacterium rhymnaticum, Geobacter metallireducens, Geobacter sulfurreducens, Geovibrio ferrireducens, Desulfoiomonas acetoxidans, Pallobacter carbinolicus, and the last one is Swanella putrefaciens. Okay, I am repeating the bacteria again. Ferribacterium limnaticum, Geobacter metalli reducens, Geobacter sulfur reducens, Geovibrio ferri reducens, desulfuromonas acid oxidants. Next one is Pellobacter carbinolicus, Swanella putrefaciens. Okay, now moving further, a short video. Okay, a short video. Okay, a ferruginous spring. Okay, in Switzerland. You can clearly see the red rust precipitates. You may have also seen at various other places also around you also. Okay, there are various places where rust is easily visible. Brown flocculation you may have also seen. Okay, and as you all know, these flocks, okay, these are the flocks. Okay, and this is an undrinkable water, and the flocks are the most probably filamentous. They are the filamentous iron bacteria. They are the filamentous iron bacteria. Now, in such an environment, bacteria are able to oxidize the soluble ferrous into sparingly soluble Fe3 hydroxides. Okay, now in this video, okay, the presence of iron and bacterial activity with the Prussian blue reaction. With the Prussian blue reaction. You may have studied in your school. Okay, now these are the iron oxides and are passed through a 0.45 micrometer membrane. Okay, if you remember this one, this membrane is used for the filtration. Okay, for the filtration of bacteria. Okay, these are special filters. Okay, this is in Leptothrix orchiacea appears as brittle yellowish tubes. Now, simply place a membrane on a paper filter soaked with a 5% of potassium ferrocyanide. Potassium ferrocyanide. Okay. Allow to incubate for 10 minutes. Soak a new filter paper with 5% HCl and lay the membrane on it. And lay membrane on it. The Prussian blue precipitate is produced, and therefore now iron is present. How? HCl induces the solubilization of Fe3, okay, and the Fe3 which is present in excess produces a blue colored complex by intervalence charge transfer. This is basically an Prussian blue precipitate. Okay, if you want to do in the laboratory, you can also do. You just need to find out the iron bacteria. Okay, the flocculation. Okay, the flocculation. Now the filter surface. Dye the membrane. Okay, પછી okay, stalks Okay, these are basically stocks. Okay, you can see the stocks present which are being produced by the iron bacteria. Okay, these are the stocks which are produced by the iron bacteria. You can clearly see the sheet covered around it. Okay, and if you observe sample directly, tube-like structures, tube-like structures. Okay, you can clearly identify this one. A sheath, you can clearly see sheath which is present around the bacteria. Okay, and what is sheath? It is an complex made up of proteins and polysaccharides which are excreted by the bacteria. Okay, now twisted stalks, see the stalks here, twisted ones. Okay, iron oxidizing bacteria are genetically diverse also. Bacterial cells. Okay, these are the various different types. Okay, again this video is by Bernard Jenny. Okay, now we will continue